Morning, everyone, and welcome to today's MPG Primer. On such a gorgeous day, we're so glad to have you all joining us both here and online. I'm very excited to introduce today's speaker. Xin Wang got her MPH from the University of Michigan, and since then has come to work here between the Broad Institute and MGH, where she's really led and participated in a huge number of different projects, including what she'll tell us today, which has been an analysis that integrates disease risk prediction and thoughts about how to best target the mechanisms of the underlying disease. She's also done a lot of work on both rare and common variants in large biobank cohorts. And very excitingly, she will be starting her PhD at the Sanger Institute this fall. So we thank her for taking the time to give this presentation at the end of her tenure here. She'll be happy to take your questions during her talk. And so please either come up to the microphone or I'll be watching the Q&A on the Zoom. So thank you so much and thank you for joining.
Thank you. So thanks for having me today and thanks for the kind introduction. So as the title suggests, we are going to talk about using individual level tissue-specific imputed transcriptome to find drug target candidates and to predict disease risk. So today's talk will consist of 4 parts. First, I will give you some introduction and background information so that we can be on the same page about why we did this project and what the framework looks like. And then I will share some results of transcriptome-wide association studies and co-localization analysis for phenotypes of interest to find potential drug targets. And then using the same list of phenotypes of interest, which are cardiometabolic traits, we will show how we constructed and tested gene expression-based risk score for disease risk prediction. And then we will switch from a phenotype-centered analysis to a gene-centered analysis, where we run Phenome-wide association studies for established drug target and explore the possibility that this method may prioritize some novel indications for approved medication. So why we are interested in gene expression? Because gene expression levels causally contribute to phenotypic variability, and this effect can be tissue specific. For example, the expression levels of the PCSK9 gene in liver cells is associated with the levels of low density lipoprotein cholesterol in the blood. So, for example, here in liver cells, there are some LDL receptors on the membrane, which is responsible for binding with LDL cholesterol and taking it into the cell, so that can reduce the level of LDL cholesterol in the blood. So there's another protein encoded by the PCSK9 gene, which can also bind with LDL receptors. And when it binds with the receptor can no longer bind with LDL cholesterol. So that will result in an increased level of LDL cholesterol in the blood. So there's a group of medications which inhibit the expression levels of PCSK9 gene so that it can free more LDL receptors for it to bind and remove LDL cholesterol from the blood. So this is an example of how gene expression causally contributed to a phenotype and why this effect sometimes can be tissue specific, because for other tissues, for example, skin or muscle, this relationship may not hold. And so what we want to do is that we want to find more examples like this, so that may provide opportunities for novel therapeutics development for other diseases or phenotypes. So, but we all know that tissue-specific gene expression is very difficult to measure at scale. So as you can imagine, it's almost impossible to take tissues from different organs at a population level. So what we did is that we leverage statistical and computational methods to impute the genetically regulated gene expression levels across different tissues. And in addition to using it as a variable that we can leverage to find drug target, the tissue-specific imputed gene expression profiles at the individual level can also be used to infer individualized phenotypic risk. And we hypothesize that this score may be more biologically interpretable than other conventional scores, because the predict unit in the score is the gene expression level. So the mechanism is clear. So what do we mean by imputation? So this is a group of methods that developed for transcriptome-wide association studies. And they usually started with training prediction models in a reference panel, such as the GTEx dataset. So in a panel like this, you will have whole genome sequencing data and a real gene expression data measured using RNA sequencing technology across the different tissues. And for example, GTEx has around 50 different tissues, and the sample size ranging from 70 to 700. So it depends on different tissues. And using the genetic variants from the whole genome sequencing data and the real gene expression levels from RNA sequencing data, you can model the relationship between genetic variants and tissue-specific gene expression levels to get some variant weight to use in the next step. So this step, normally the method will use penalized regression model such as ElasticNet, and it will select features and also adjust the scoring weight for the calculation. So for example, one gene, there may be like 10 genetic variants that impact the gene's expression level. And we will include the 10 variants into the model, and then the model will give an adjusted weight for each of the variants. And then by using the linear combination of those weighted variants, you can calculate a value for the gene expression. So apply those models to an independent population biorepository, such as the UK Biobank, where we have 480K participants with genetic data, but we don't have gene expression data there. So we are using the weights and then combine with the genetic variants data in the UK Biobank. We calculate or predict the genetically regulated gene expression levels across different tissues. So you can think of the predicted values as a polygenic score of gene expression. So after the imputation, we have 480K samples in the UK Biobank, and here is the baseline characteristics of our sample. And we are interested in 6 phenotypes, including 3 diseases and 3 quantitative traits. So the disease include atrial fibrillation, coronary arterial disease, and type 2 diabetes. And quantitative traits include QT intervals on ECG, which is a parameter measuring the time of ventricular contraction and relaxation, and is highly related to atrial fibrillation. We are also interested in low density lipoprotein cholesterol levels, which is a parameter highly related to CAD. And then also this HbA1c level, which is a measurement, which is a parameter measuring the blood sugar control ability over the last 3 months. So it is a parameter highly related to type 2 diabetes. And for these 6 phenotypes, we identify their biologically relevant tissues using domain knowledge. That means we have our MDs and supervisors to review the list of tissues and identify their biologically relevant tissues. So at the time of this analysis, we were aware of some computational methods that can prioritize biologically relevant tissues for phenotypes using summary level statistics. We tried a couple and we found that because gene expression profiles across different tissues are often highly correlated. So some methods may not distinguish well between tissues that are truly relevant to a phenotype and tissues that are not truly relevant, but highly related to truly relevant tissues. And because of this, we end up using our domain knowledge to identify those tissues. But we are also aware that recently there are some new computational methods come up that can address this correlation issue better. So this may be a new research direction for our plan, of our future plan. So, and then in the last column is the number of imputable protein coding genes in each of the tissue. So this number is essentially a function of the sample size in the gene text training panel. And so, for example, in GTEx, the adipose tissue has a larger sample size, so it has a better statistical power and then more available prediction model. And for liver, because it has less sample size, so the high quality prediction model, the number of high quality prediction model is less. And then comes to our first analysis, which is the joint analysis of TWAS and co-localization to prioritize druggable genes for phenotypes of interest. This analysis consists of 3 steps. First, we run these transcriptome-wide association studies in biologically relevant tissues for each phenotype. So for example, for AFib, we evaluate the association between AFib and all genes that can be imputed in the true heart tissues available in GTEx data. And then we will have a list of significant TWAS signals, and we process this list of associations using a co-localization analysis. I will go to more details about why we need this additional step in later slide. So the third step is that for gene tissue phenotype combinations that's supported by the first 2 steps, we will annotate their therapeutic targetability. So which means we find genes that encode actionable human proteins, and these proteins are drug targets of approved or clinical trial stage medications. For this step, we use a previously published data set to do this. So why we need a joint analysis of TWAS and co-localization? So the short answer is that TWAS sometimes will give you a false positive result because of linkage disequilibrium. So what does that mean? Here is an illustration from the co-localization analysis paper. And in panel A, so panel A is the real world TWAS result from the UK Biobank body height data. And each dot on that panel, so each dot on that panel is an individual gene. And this is not the significant line. This is the cutoff because this signal is too high. So the significant line is here. If you can see that a band red line. So everything above that line means they are significant in this TWAS analysis. So you can see there is a cluster of significant genes centered around this ZBTB38 gene. And the panel B, in panel B, the authors replicate these similar patterns using a simulated data set, assuming that there is a causal variance within this ZBTB38 gene. So when the causal SNP is in this causal gene, other genes still significant and center around this top signal. So in panel C, they regress out the causal SNPs from the simulated phenotype data. And as you can see here, the entire cluster just disappear. So which means the reason why those other genes are significant before is not because they have a causal variance within their region. It is because they are close to the true causal gene. So let's zoom in a little bit at the variance level, explaining why this is happening and why we need a co-localization analysis. So this is also an illustration from a previously published paper. So these 4 panels are association study results from 4 different phenotypes. So for example, in the first column, on top is the log-long plot from association studies of the LDL phenotype. And this is the cluster of genetic variants. So each dot here is a genetic variant. This is the cluster of the variant signals that are significantly associated with the LDL phenotype. And on bottom is a log-long plot from an association study for the gene expression level of the FRK gene. So here is to associate variants with LDL phenotype. And here is to associate genetic variants with gene expression levels of FRK. So what the co-localization analysis does is that it estimates the probability that these 2 signals are shared. So as you can see here from the location, the peak of these 2 signals are actually different. So that's why the analysis gives a PP3 of 99%. PP3 means these 2 signals are not shared. So and a real case is the second column, where on top of it, it is also an association study for the TC phenotype, which is another cluster. And these are the cluster of variants that are significantly associated with this phenotype. And on bottom is the cluster of variants that are significantly associated with the SDC1 gene expression level. So as you can see, based on the location, these 2 signals are likely to be shared. So that's why the co-localization analysis gives a PP4 probability of 82%, which means these 2 signals are likely shared. And because of this, so the variants that impact phenotypes are the same as those variants impact the gene expression level. So it's more likely that the variants impact the phenotype by impacting the gene expression level. So this is a case that's a scenario that we want to have by using the co-localization analysis to process the TWAS results. And using a probability cutoff, you can prioritize high confidence TWAS results from the first step. So here is the result of our entire analysis. And each panel here represents 1 phenotype with one of its biologically relevant tissue. So for example, this panel is LDL cholesterol in the liver tissue. And each dot on each panel represents an association between that phenotype and the expression levels of a protein coding gene in that tissue. And all the signals that's supported by both TWAS and co-localization analysis and potential therapeutic targets were annotated using their gene name. And genes on the left-hand side is negatively associated with the phenotypic risk, and on the right side is positively associated. So in this analysis, we identify in total 5 therapeutic targets that have an approved medication targeting it. And on top of the gene names, we show the co-localization probability from our analysis. So for example, the ANGPTL3 expression levels in the liver is positively associated with LDL cholesterol level. And in fact, ANGPTL3 inhibitor is approved as a lipid lowering medication in the market. Another example is the IL-6R gene. So IL-6R inhibitor is approved to treat a group of autoimmune diseases. And atrial fibrillation is not in the list of approved indications. However, inflammation has long been hypothesized to be a risk factor for cardiovascular diseases, including atrial fibrillation. So we think this may be an potential drug repurposing opportunity for atrial fibrillation. So and then come to our second analysis, which is gene expression-based risk prediction. So using genes that or gene tissue pairs that's identified in the previous TWAS, we constructed two gene expression risk score for each phenotype. The first score consists of all genes identified in the TWAS, and the second score consists of potential druggable genes identified in the TWAS. So the predictors in the second score is a subset of the predictors in the first score. It just the second score only contains potential druggable genes. And to compare our gene expression scores to a more conventional prediction approach, we also calculate a polygenic risk score for each of the phenotypes. And we include all genetic variants in genome-wide association, in a previous genome-wide association study to calculate the score. So here is the overview of our analysis. So for the disease phenotypes, we train the scoring model or like obtaining the scoring weight in that derivation data set, which is the prevalent data set. So we identify people who were diagnosed with a specific disease before enrollment as cases, and other people as controls. And we include all gene tissue pairs into a model to get a scoring weight. And then we calculate the score in a validation data set, which is the incident disease data set. And we remove all individuals that were prevalent cases, and we treat incident cases as cases and other people as controls. And then we use a Cox model in the validation data set to evaluate the association between the calculated score and the phenotype. And because this framework considered the two periods of the disease risk, so they were considered relatively independent. And then for quantitative trace, because there is no incident outcome, so we simply randomly split the complete data set into that derivation set and a validation set. So it's a 30% and 70% split. To calculate the gene expression risk score, so in the derivation data set, we input all gene tissue pairs that's identified in the TWAS into an elastic net regression model. So for example, gene I in tissue J is significantly associated with AFib in TWAS, and gene I in, say, tissue B also is a signal. So we will input all those gene tissue pairs into a penalized regression, and the model will select features and adjust the weight to calculate the score. And we are applying those adjusted scoring weight in the validation data set to calculate the transcriptome risk score. And as I said, for disease phenotypes, we model the incident outcome using a Cox proportional hazard regression. And for quantitative trait, we use a linear regression model. We have a similar framework for the polygenic risk score. We're also in the derivation data set, we input, we use the GWAS summary statistics from previous study and input the summary stat into the PRSCS algorithm, which this algorithm will also adjust the variant weight to avoid overfitting. And then we apply this scoring weight in the validation data set to calculate a polygenic risk score. And it's again, we test the disease and quantitative trait differently using different methods. So in the validation data set, we observe that gene expression-based risk scores and polygenic risk scores are complementary. So here is an example from the type 2 diabetes analysis. And if we put the gene expression risk score in their individual model, they are significantly associated with type 2 diabetes risk. And the same for the genome-wide polygenic risk. And if we put a gene expression risk score and PRS in the same model, they remain significantly associated with type 2 diabetes risk in the validation data set. And we observed the similar qualitative results for all the phenotypes we tested in the validation set. We also evaluate the risk discrimination and stratification ability of those scores. So for disease phenotypes, we divided the sample into three different groups. The red line here, the blue line here represents the lowest risk group, which consists of individuals in the lowest 10% of the risk score distribution. And the red line represents the high risk group, which consists of individuals at the highest 10% of risk distribution. And in the middle is the remaining samples, which is 80% in the middle. So as you can see, we observe a clear separation between different risk groups for the cumulative disease risk. And then for quantitative trace, we also divided the sample into three different groups using the same cutoff, the lowest 10%, highest 10%, and the middle. And we compared the quantitative trace between different risk groups, and we can see a separation between groups. So this is our last analysis, which is a Phenome-wide association studies for established drug targets. So by established drug targets, we mean genes that encode single protein targets or non-drug binding subunit of protein complex targets with approved medications. So we extract this list from a previous published data set, and then we impute those genes, the expression levels of those genes in whole blood. We chose whole blood as a proxy for tissues relevant to different drug tissue combinations, because we are going to test a large number of genes here and a large number of human disease here. So whole blood, because it is a generic tissue and its expression profile is moderately correlated with other tissues in the GTAX panel. So we use it as a proxy for this analysis. And to scan the human phenome for each of the genes, we will need a large number of phenotype definitions. And to achieve that, we leverage the Phenome-wide association study and the FICO mapping algorithm. So what is TWAS and FICO? TWAS is a research approach that investigates the association between a variable of interest. So in our case, it is the imputed gene expression level and a wide range of phenotypes or traits. And the FICO algorithm is a mapping method that enables us scalable phenotyping by using ICD codes from the EHR data. So for example, here is a list of the ICD-10 codes, and all of them represent type 2 diabetes with different complications. And the FICO mapping algorithm will group them together to create a single phenotype, type 2 diabetes. So comparing to running association studies for each of the individual ICD codes, which are essentially represent the same phenotype, running an association of the group, the phenotype, will give you a better statistical power in TWAS using EHR data. So we tested associations between 541 human diseases and 94 established drug targets that can be imputed in the whole blood tissue. And using a significance cutoff, we identify 15 significant signals from the association study. And again, we further process the result using a co-localization analysis for the reasons we just described. And 11 of the 15 associations had a non-zero co-localization probability, and 3 of them have a co-localization probability greater than 0.5, which is the cutoff we used for co-localization analysis. Here is the result prioritized from our TWAS study. And the first column is the names of the gene that we tested. And the second column is the phenotypes encoded by B codes using ICD code. And here is this column is the effect size from the association studies. And the last column is the co-localization probability. And we would like to highlight the example of the JAK2 gene. So blood expression levels of the JAK2 gene is positively associated with myeloproliferative disease risk. And in fact, the JAK2 inhibitor is approved as a medication to treat myeloproliferative disease. So we think this signal is an on-target replication. And the JAK2 expression is also significantly associated with inflammatory bowel disease risk, which is not an approved indication for this medication. But this signal is supported by previous literature and ongoing clinical trials. So we think this may be a signal that can be a potential drug repurposing opportunity. A similar example is the on top of the IL-6R gene and heart disease, which again supported the hypothesis that inflammation in the heart disease may be relevant to cardiovascular disease. So our results need to be interpreted in the context of design. First, we use the UK Biobank as our study sample, which is predominantly consists of European ancestry samples. So this may limit the generability of our result to other ancestral groups. And we also note that the prediction models that we used to impute transcriptomes normally only contain these variants in their prediction, which means these methods using genetic variants that's close to the genes that they want to impute in the model. And we know that some genetic variants or regions far away from the gene may also impact a gene's expression level. So we anticipate future methods that leverage transvariants can improve the prediction quality of this method. Gene expression is a multifactorial variable, so some genes may be very sensitive to environmental factors. So for example, your dietary style may impact some genes expression level. And our methods only capture the component of gene expression that can be explained by genetic information. So the take-home message from this talk is that our study provides a framework for using individual level tissue-specific imputed transcriptome in drug target prioritization and disease risk prediction. We show that gene tissue trait association inference prioritize the druggable genes for phenotypes of interest. And we also show how to construct and test the gene expression based risk scores, and they may be more biologically interpretable than methods for which the predictor unit, their mechanisms underlying the disease may not be well understood. And lastly, we also show the Phenome-wide scans for its established therapeutic targets may help identify potential null indications for approved medications. I would like to thank my supervisors for their support of this project and thank my colleagues for helping me and working with me on this project. So with that, I'm happy to take any questions. Thank you.
Thank you so much for sharing all of those just very impressive approaches and results with us. If anyone in the room has a question, please feel free to come up to the microphone, or if you're online, feel free to type in the Q&A. I was wondering, you chose relatively prevalent conditions. Do you have, it seems like there's an interplay between the genetic contribution to disease risk, your total sample size, and your affected sample size. Do you have any gestalts on kind of the what the minimum disease prevalence and genetic contribution might be for a disease to be tractable by this method?
Yeah, that's a great question. So for the phenotype-centered analysis, we do have those like relatively common disease. But for the phenome-wide association study, because we use like a large number of definitions, and some of them are less prevalent disease. But we applied a cutoff of 20 cases at least for the analysis. And we did observe that for like less prevalent disease, you probably have like less statistical power for the test. So yeah, we don't have a like clear answer about like above what number of cases you will see a signal, but we did observe that if you like more cases is definitely better, but like it can still work at some like low case number scenarios. Thanks.
Another question I had was when you think about imputing gene expression, there's obviously the risk for more perturbed gene expression with time after diagnosis, especially for some of the cardiac or other disorders that might have progressive fibrosis in the organ. Do you think your model is successful because you are imputing the pre-disease transcription profile? And is that therefore a potential advantage over looking at kind of the disease state transcriptome? Or I guess, was there any ability to compare in the disease state, the imputed versus the kind of progressive differences in transcriptome?
Yeah. I think that is a like great idea. And we also think about like it would be great if we can have like the gene expression level before, after the disease so that we can compare. So, but in the method we use, because those models, they really train on a reference panel. And so for example, GTAX, I think most of the samples, they are probably healthy samples. And we don't have, I don't think we have the data about their like disease diagnosis. So, and also because like the model, it's like it models the relationship between germline genetic variants and the gene expression. So it may capture the component that can be explained by your genetic information, but we are also very interested in like how the change like before and after disease can have some more information in terms of drug target prioritization or predictive disease. Thanks.
Well, thank you so much for your talk today, and congratulations on your upcoming graduate school.
Thank you so much. Thank you.